This is screen capture video unit 7.1, and unit 7 has to do with energy transformation, cell movement, and plasma membrane. And to start off the discussion, if you read different texts that cover this type of material, a lot of times there's a discussion of the laws of thermodynamics. The first law of thermodynamics states that energy is neither created nor destroyed. Technically, many would say that's the energy law. When you're talking about thermodynamics, it would actually be matter and energy are neither created nor destroyed. But we're focusing on the energy for this unit. Some might point to a thermonuclear reaction and point to the fact that the matter is being destroyed in a sense, but when you take in the energy and matter, it fits the law, because even though the matter is being used up, it's being transformed in a sense into a tremendous amount of energy. In the illustration that I pulled off the internet, this is supposed to be illustrating a coal-burning power plant. It looks like in Georgia. If you consider coal, that would be from plants and animals that lived many years ago under the right conditions, were turned into coal. And going back just a little bit further than that, if you consider the many plants involved, those got their energy from the sun. If you're talking about any animals, they would have consumed animals that ate plants or consumed plants. So ultimately, the sun is the energy source for any of your petroleum substances, including coal. Well, every time that you have an energy transformation, energy is being transformed, and in a sense it's being lost to heat energy. Now when you're burning coal, you want heat energies, but some of the coal burning would produce light that wouldn't be used up in the coal burning power plant, but the heat would be. The heat would cause steam to increase in temperature, cause it to change state, and go from liquid to a gaseous state. In the gaseous state, it would create greater pressure, because generally any time that you change state of any type of matter from liquid to a gas or solid to a gas, and especially if you do it quickly, you'll create a lot of pressure. That's the idea with explosives. So with the steam increasing pressure, it runs the turbine, and so you're going from kinetic energy with the light from the sun to potential energy stored in chemicals of the coal, to kinetic energy with increasing the temperature. Heat energy is kinetic energy. Moving the turbine would be a type of kinetic energy. That turning of the turbine would turn an electromagnet within another electromagnet. Here you have more kinetic energy, and that produces electricity, and that goes through transformers to up the voltage to many thousands of volts, so less energy is lost as it travels miles through the power lines, the transmission lines. And then when it gets to a home or business, the voltage is stepped down, the amperage is stepped up, and then the electricity is run to the house or the business, and it would go to produce light, in some cases heat, but even if it was producing light or running equipment, you're going to have some energy that's lost as heat. So you can keep track of all that energy, even that which is producing a type of energy you don't necessarily want to produce, whether it's light energy or whether it's heat energy. You can keep track of that and measure it. The second law of thermodynamics states that no energy transformation is 100% efficient. I alluded to that before when I stated that in any energy transformation, you're going to have some lost as heat. You're going to have heat produced, even if you're not necessarily wanting heat produced. Usually that happens. Another way of stating the second law of thermodynamics is to state that in any system, closed system, entropy increases, and entropy can be thought of as disorder. And so if you think about an apple, you set an apple off to itself, over a period of time it moves toward a disordered state. That is the natural flow of things. That's the natural flow in the universe, is to move towards greater disorder. And the way that disorder is stopped or slowed down is to put energy into the system. And that happens with Earth, with the sunlight that shines on the Earth. There are different types of reactions. There are what are called endergonic reactions. When you think of ender, you could think of into. Energy is going into the reaction. And I have a graph, a chart here that shows an endergonic reaction. So as the reaction progresses from left to right, the energy that is needed to get the matter to react is sometimes referred to as the free energy. I won't get into the delta G, but to go from the beginning chemicals to the end chemicals, the products, there will be energy that's required to move to that point. And this would be called an endergonic reaction because energy is required to be put into it. The opposite would be an exergonic reaction. If you think of exit, it's out of, and that's what's happening in an exergonic reaction. The energy is being released, so you're going from a higher energy state to a lower energy state, and energy is being released. And eventually, we're going to be talking about cellular respiration. And in cellular respiration, I don't want to get too far ahead of myself, but I'll talk about those a little bit later on. You can think of those on your own. Adenosine triphosphate, sometimes referred to as ATP for short, is a very important chemical when it comes to chemical reactions and energy transformations in cells. Whether you're talking about bacteria, which are the simplest creatures on the face of the earth, or human beings, which are the most complex. We are told that all those organisms in between, including the two groups, use ATP and are constantly producing it and constantly using it. I have the structural formula for ATP. Here you can see there's the adenosine, there's the sugar group, and then there are 3 phosphates. That's where you get the triphosphate. First, second, and a third phosphate group. And it doesn't show it here, but between the phosphate groups are high energy bonds. So every time you pull off a phosphate group, energy is released. And it takes energy to put a phosphate group on. A lot of times there's a squiggly line that's shown between each phosphate group. And I do show that in this way of writing ATP. It's nitrogenous base, happens to be adenosine here, the sugar, and then 3 phosphate groups. And between the phosphate groups, I show those squiggly lines, which means high energy bond. Now, it's possible to take the 1 phosphate group off of ATP and to get ADP. ADP stands for adenosine diphosphate, diphosphate meaning the 2 phosphate groups. You can take off that second phosphate group and you're left with AMP, which is adenosine monophosphate, just 1 phosphate group. And biochemists have measured the energy in those and know how much energy is released with each phosphate that's pulled off. Quite a bit for the first one, a little bit less for the second one. Putting phosphates on these molecules, whether it's AMP or ADP, or removing a phosphate, is referred to as phosphorylation. There are different types of phosphorylation. Sometimes phosphorylation will occur because of energy that's released from the direct breakdown of a carbohydrate or some other organic molecule. That's called substrate level phosphorylation. Sometimes the phosphorylation is linked because of movement of electrons, hydrogens across a membrane. Then it's called chemiosmotic phosphorylation. And there's even photophosphorylation when we get into photosynthesis. So different types of phosphorylation activities, but it has to do with adding phosphates or taking away. And usually we'll talk about the phosphorylation where phosphate groups are being added. When we're talking about energy transformations, especially with cells, enzymes become very important. Enzymes are referred to as organic catalysts, and their function is to lower the activation energy. They don't necessarily speed the reaction up, but they allow it to happen with less energy. And if it wasn't for the enzymes in our cells and outside of our cells, we wouldn't be able to survive. We wouldn't take in enough energy in a given day to survive. So our enzymes help us tremendously to lower the energy that's needed for our body to function. I suppose some people might want some of their enzymes to not function so well, so they used up more energy, but that's not generally a problem in life in general. The structure of the enzyme, there's always a protein portion. That's usually the larger part of the enzyme. That's referred to as the apoenzyme. And then there's some kind of a non-protein portion that's called the cofactor. And sometimes the cofactor is organic in its nature. It's called a coenzyme. Here it seems that there's a cofactor and a coenzyme. Generally, the coenzyme is a type of cofactor if it is present. A good example of a coenzyme would be vitamins. If people don't take enough vitamins or consume enough vitamins in their diet, what they consume, they'll have deficiency diseases. It's because in some cases they don't have the coenzyme to go into the makeup of the enzyme to carry on certain chemical activities in and around their cells. The cofactor, along with the apoenzyme together, make up the entire enzyme or the whole enzyme, sometimes referred to as a holoenzyme. Somewhere on the enzyme, there will be a location, a physical location where chemical activity occurs. It's the chemical activity that the enzyme is helping to occur at a lower energy level. Some enzymes will have one active site. Some will have many active sites. This is just showing one active site. And that is where the substrate is acted upon by the enzyme. The substrate is the chemical that's acted upon by the enzyme. And sometimes there's more than one substrate, sometimes just one substrate. Sometimes the substrate comes in, it's broken down into smaller parts. Sometimes you have two smaller parts that come in and are put together by the enzyme. And I mentioned the organic catalyst. Catalyst in that it's not used up in the reaction, but it can be active again and again and again as long as it's not destroyed or denatured. And it is organic in nature. It's made up of organic molecules, protein, and sometimes some kind of vitamin, which is also organic. Sometimes cofactors of metal. Metals can be very important in chemical reactions, helping those to occur. I talked about denaturing of proteins in an earlier unit, and denaturation is an important thing with enzymes because they're made up of protein. They are subject to denaturing, and you can denature proteins and thereby enzymes by high heat. In this illustration, it's showing heat above 40 degrees Celsius. That would be true of a number of enzymes in the body. You get above 40 degrees Celsius, and a number of enzymes will start to denature. And what they're trying to show here in this illustration is by heating up this enzyme, the shape is changed. Notice how it's elongated, and the active site is changed. So it no longer will allow the substrate to come in and physically fit at the active site. Many denaturation events are permanent. Some are reversible, but usually they are permanent. Besides high heat, high salt concentration, and pH extremes will denature enzymes, or can denature enzymes, because they do affect this protein structure, which is very specific. I'll continue with talking about enzyme inhibitors in the next video.